Thank you very much for this very kind introduction. Thank you also for inviting me here to talk at this symposium, which is really a very unique experience, getting together scientists from very different areas. The subject of my talk was already very nicely introduced yesterday by Professor Andreas Hense. The title is Exploiting Genomic Information to Understand Desiccation Tolerance in Selected Plant Species. So, what are these selected plant species? These selected plant species are resurrection plants, those which we heard about yesterday already. And now I would like to introduce to you the plant which we investigated quite a lot. It is also a plant from South Africa. It is called Curating plantageneum. And you see on the slide the plant in the three extreme physiological stages, namely going from left to right in the fully physiological, well-hydrated stage, in the complete desiccated stage, and this is far below 10% of water left in the tissue. And on the right, you see the recovery, and this picture is taken about 24 hours after adding water to the fully desiccated plant. And these processes, namely the process to go from the fully hydrated to the desiccated plant, and again going back to the recovered plant, these processes, they really demand very specific genetic programs combined with expression of new proteins and also with specific metabolites. And what I would like to do this afternoon is to introduce to you some of our findings while studying these different processes. So this plant has not only the advantage that it can recover from complete desiccation, but it also has a disadvantage, namely that it is not really amenable to genetic transformation because it has an octoploid genome. So it's difficult to make mutants. Therefore, we have been trying to take a different approach. A colleague of mine, and I think some here in the audience know him, Eberhard Fischer. He is a true botanist, and he has provided us with some plants which are very closely related to the Curating plantageneum, the two Lindernia species, which you see here on the slide. So you can see from the flower morphology, going from left to right, that the flowers are very similar. So that sort of indicates their relationship. So why did we also include these plants in our studies? Namely, because the Lindernia supracemosa, written here in red. And if you want to follow my talk, you will always see those plant names written in red, which cannot desiccate completely. So you don't have to memorize the names. Whereas you will see the names written in green when the plant is desiccation tolerant. So here, this slide shows you these three species. Curating, I just introduced to you, and I showed you the desiccation tolerant phenotype. Then a closely related plant, Lindernia supracemosa, which occurs in marshy areas and which is not desiccation tolerant. And then that was a big surprise when we started to study this plant. It's Lindernia brevidens, which actually occurs in the rainforest. So in its natural life, it never experiences desiccation tolerance. However, when we tested it in the lab, it had features of desiccation tolerance. So you might call it a sort of bit of an intermediate phenotype, if you would like so. And I will talk more about the comparison of these three species. Before doing that, I will try to summarize in the following slides what we wanted to do, what is our objective, and just a sort of broad overview, what have we found. So why do we study these plants? What are the benefits? First of all, and that is our primary interest, is the identification of mechanisms which can be perhaps translated to withstand water deficit in agriculturally important plants. Then, and we heard quite a bit about that yesterday already, it is possible to discover interesting metabolites which may have beneficial properties in very different contexts. And then also thirdly, and I will show you a short slide on that later, is that the biochemical studies might indicate how we can get a new perspective on molecular reactions, namely understanding metabolic reactions in a non-aqueous cellular compartment. And to the third aspect, we know the least. But this is something we would like to study also to go into the future. First of all, now let me come to the summary. What do we know? And it's a very rough summary. So we have studied the resurrection plant, Cladostigma plantageneum, which you see here in its natural habitat, left the fully hydrated plant, and then on the right, you see the plant just before the rain starts. You see some other plants are already recovered. You can see here in the desiccated state. Perhaps one other feature which I would like to attract your attention to is here, and which you can also see on this slide, but maybe even better. Oh, I can't go back. Okay, doesn't matter. The Cladostigma plant retains most of its chlorophyll in the desiccated state. So maybe you noticed that already on the previous slides. So what did we find? What happens roughly during the process of desiccation, dehydration? So we find major changes in the carbohydrate metabolism. And interestingly, and I will not come back to that, is that we found the high accumulation, really high, of the uncommon sugar octulose, which is a CH sugar, whereas in the fully hydrated plant. And then this is somehow converted to sucrose in the desiccated tissue. And the accumulation of sucrose, as we heard already yesterday, is a sort of common theme in desiccating organisms, from microorganisms to higher plants. So major changes in carbohydrate metabolism. Then we find induction of a broad array of proteins, which I will not go into much detail. And a subclass of these proteins are the so-called stress proteins. And the class of these are the so-called late embryogenesis abundant proteins. I will talk about them later. But also metabolic processes which require the protection, photoprotection, because as I said, it retains its chlorophyll. So it still absorbs sunlight, and you still have the accumulation of oxygen radicals here. Also, an aspect about which I will not talk about much is the cellular structures are affected in this dehydration process. Namely, if the plant, the cells start to shrink, they lose most of the water from the vacuole. So there's quite a high mechanical tension, and this requires modifications in the cell wall of this plant. And we did some experiments to that. The next slide is quite a busy slide, but it also summarizes, again, some work which we did in collaboration with a group in Luxembourg, namely that we tried to put together the induction of genes, the accumulation of messenger RNAs throughout the 4 different stages, which you can see again from left to right: fully hydrated, partially dehydrated, desiccated, and rehydrated. We included partially dehydrated in our studies because we wanted to see where's the switch to the desiccation metabolism, or you could even call it as now to the desicome. So, and really most processes which you know very well from your school even are affected. I mean, it's the photosynthesis, photorespiration, cellular metabolism, etc. I will not go into detail here. And I would like to include another different consideration in our concept of the studies. Namely, what you see here is a series of dehydrating wheat grains. So from the immature wheat grain on the top left of the slide, going to the bottom row where you see the dry seed. And you could present such pictures more or less from many higher plants if you consider the seeds. So what happens in these seeds? They also lose water, and they also go to a desiccation tolerance. And this is something which you take for granted. Perhaps you don't think about it so much. And what I've written here on the headline is how do these seeds prepare for the metabolic arrest? Because if you know, you can go to a shop and you can buy dry seeds and they can sit there for months or maybe even years. You take them home, you add water, and they start to germinate. So they're still alive. The seeds are not dead. And just two main points, which I would like to draw your attention to, is that the phytohormone abscisic acid, or short ABA, regulates this seed maturation process as one of the main regulators. There are many more. And also what was observed many, many years ago is the synthesis of these so-called late embryogenesis abundant proteins, or in short, LEA. They were just termed LEA because one didn't know what they do, and so they were just described according to their appearance, late embryogenesis, during the late embryogenesis of these seeds. And as I would like to try to convince you later, these late embryogenesis abundant proteins, the LEA proteins, are a major determinant of desiccation tolerance. And they are not only a major determinant in the desiccation tolerance of the seeds, but also, as we will see shortly, in the resurrection plants. So what makes a resurrection plant different from a non-resurrection plant? And I will just show you 3 slides now, introducing a slightly different resurrection plant, namely the small grass, Oropatium tomeum, which originated from India. Again, you see here going from the left to the right, fully hydrated, desiccated. Here you also clearly see that it retains its chlorophyll, and then you see the recovered plant. So this plant really caught interest in a collaborative group in the US, now in Michigan, Rob van Buren and colleagues. Why did they get interested in this? Because we published some time ago that this plant has a diploid genome. Remember in the beginning, I told you that the Cratostigma has the octoploid genome and is therefore difficult to study on the genetic level. However, this plant, we had shown that it's a diploid genome and it has a very small genome. So what we did is, in collaboration with Rob van Buren and colleagues, the genome of this plant was sequenced as one of the first genomes of the resurrection plants. So what did we find from this genome sequencing? And one aspect is shown on the next slide. What you see here on the slide, maybe I hope it's clearly to see, but anyway, you see a part of the genome of this small grass of Oropatium on the top. And then this part is correlated to homologous, let me call it homologous parts of other plants, namely other grasses. And these include also cereals like rice, like sorghum, etc. And the genome is painted with the different colors. So we have the blue color, we have the orange color, and we have the sort of green color. And the blue color contains the protein encoding genes, and the orange are regions in the genome which are so-called interspersed, which don't have the capacity, as far as we know, to code for proteins. So you could see that this part of the genome aligns quite well with parts of the genome of these cereals and other grasses. So this was a very interesting finding. I mean, the difference is indicated here with its orange boxes, namely that there's much more so-called interspersed DNA in those cereals, for instance, which have a much larger genome. But the arrangement of the genes is very similar if you make this comparison. So this was actually quite an important finding. And then we looked into more deeply into the protein coding regions. And I will tell you now two examples. First, I would like to focus on these layer proteins, layer genes, which I have already alluded to. Namely, I have told you that these are major components of the desiccation, of the seed desiccation behavior, and also of the leaves of the resurrection plants. So what you find in seeds of the resurrection plants, in terms of accumulation of proteins, you find in the leaves of these resurrection plants. So what you could say is that there is a loss of control of synthesis of layer proteins in the resurrection plants. So as soon as, for instance, wheat seeds start to germinate, the layer proteins disappear. However, the capacity to induce layer or to express layer proteins during the onset of dehydration remains in the leaves of these resurrection plants. So this is quite an important finding, many years actually ago observed. So we can say that the desiccation tolerant plants have a very characteristic layer gene expression profiles. I told you that we have sequenced this small grass, the genome sequence, but we continued this collaboration with Rob van Buren, and we also sequenced all the 3 plants which I showed you in the beginning, of which we made the comparison. So this octoploid Craterostigma genome was recently sequenced, and also the 2 Lindernea sequences. So we could go more into depth to see what happens with these layer proteins in these different genomes. And that is something I would like to tell you now in the following slides. First, still a little bit more information on the layer proteins. So as I told you, they are the most abundant group of dehydration-induced proteins in Craterostigma plantageneum leaves when you dehydrate them. And interesting is that these layer proteins, if I talk about layer proteins, it's not a homogeneous group of proteins. I mean, they are subdivided into different classes, etc. And although they have been discovered in the seeds about 40 years ago, the correct biochemical function is still not really known. But what brings them together in the group of layer proteins is that they have some very characteristic biochemical properties. Namely, they are very hydrophilic. They have a lot of hydroxylated amino acids in their composition. And if you go to an aqueous situation, they have a sort of unstructured configuration. And they're also boiling stable, which has to do with the hydrophilicity. Without going into detail here, from several labs, there's some evidence that these layer proteins can make a contribution that they have a protective function under water stress conditions. So, and my hypothesis is that during the evolution of this extreme desiccation tolerant phenotype, the abundant expression of layer proteins has to be somehow achieved. And I would give you an insight how this, one of the possibilities, how this can be achieved. There are 2 possibilities actually. And the expression of these layer proteins should also be very tightly regulated during the dehydration process. First, 2 slides on the structure, just to tell you what I've, or show you a little bit more what I have just summarized. If you go here from the left to the right, you see the part of a layer protein, a cartoon, in an unstructured, in an aqueous situation. So it's a non-structured protein. However, if you go to dehydration, then you can see that it assumes either alpha helix or also other very specific structures. This was actually when we first wanted to determine the structure of a layer protein. If you just take the pure DNA and protein sequence and you make a prediction, you will predict a lot of alpha helices. But in a collaboration with a group in Regensburg at that time, we tried to determine, to find these alpha helical structures. We couldn't find them. So what was wrong? I mean, it was wrong that we did not look into the dehydrated state. And now this has been confirmed, and not only from plants, but there are also some desiccation tolerant nematodes. And most interestingly, also these nematodes make some layer proteins during the dehydration. And what you see on this slide is exactly what I've tried to show you in the previous slide, that these layer proteins assume a structure only during the dehydration. If you go from top to bottom here and go to the right hand side, you see that the water content decreases, and indicated here in the red are the alpha helices, which then are assumed during the dehydration process. So I think this is also very interesting. I told you they are very hydrophilic proteins, and one of the hypotheses is that these hydrophilic proteins can in part substitute the water in the desiccated cell and maintain structure of proteins of nucleic acid as it is required then for the survival. So this is one aspect, but it could also be a very interesting aspect if you think of chemical reactions where you have this change. How do chemical reactions change in such an environment? But this is an open question, and I cannot go into more details because we don't have more details. So I would come back now to the layer proteins, so where we have some answers, namely when we make this comparison between the desiccation tolerant and non-desiccation tolerant plant species. Here again, you see the two tolerant ones written in green and the one in red. So how did we go about it? We aligned the genome parts and we tried to classify these layer proteins, and we tried to identify regions in the genome with homologous genes via sequence comparisons. And we looked at the organization of these layer proteins in the genomes of these 3 plants. And we also determined the expression during different stages of dehydration, rehydration in these plants. And the next slide is a relatively busy slide, but I just want to give you at least one experimental piece of evidence. Here you see the expression profiles of the layer genes in the 2 Lindernia species, brevidens, desiccation tolerant, and this is the one here on the top row. You see that we compared to the Lindernia supracemosa on the compact bottom row, and 7 stages of dehydration, rehydration are compared, but just without going into detail. So you can analyze this, and I will come to the conclusion from this analysis in a minute. But what you can even visually see is that on the brevidens row, you have many more blue spots and also darker spots. So that means we have a higher expression of subclasses of these layer proteins. On the top of the slide, you see these different groups of layer proteins. I told you it's not a homogeneous group of proteins. So what can we conclude from these different comparisons? Now, in a very simple sentences, namely, what we find is that subsets of layer genes are induced by dehydration in all 3 species. Disregard whether it's tolerant, or non-tolerant. Also, for instance, if you take Arabidopsis, if you take wheat or barley, they all will synthesize some Lea proteins during a dehydration process. So that is one of the messages. So, but are there differences? Yes, there are differences. Namely, selected gyasins are much more abundantly upregulated in the desiccation-tolerant species, Craterostigma and Lindernea brevidens, if we compare it to the non-desiccation-tolerant. And when we looked into more detail even of these subclasses, which seem to be, in the first glance, common, we also find that this is not identical always, but that the expression analysis across these different species suggests that during evolution, there has been also a neofunctionalization of the genes after duplication events. Because in the genome of the Craterostigma and the brevidens, we find more Lea proteins, and they have, or Lea genes, sorry, and they have arisen by genome duplication. That is one of the evolutionary processes which contributes to the stable desiccation tolerant type. But we will, in a minute, also get to know another way. So, namely, how do we get to this high expression? Remember, you saw the different blue colors, dark blue, lighter blue. So that means more or less of the same gene or gene product present. And in addition to the duplication of certain gene regions in the genome, we also find specific gene expression patterns which are determined by the regulatory or promoter regions. And we made a comparison, and I will show you one example, not only of the coding region for these Lea proteins, but also of the regulatory region of the so-called promoter region. And here you see a cartoon. Again, we take the same gene, the homologous gene, let's say, from the three species which we compare, Craterostigma, Lindernea brevidens, and Lindernea supracemosa. And if you just look to the right, and you can see the red and the yellow motifs. These are very short nucleotide regions. These are specific promoter regions, and they determine whether a gene is expressed under dehydration or not. And if you go to Craterostigma, you see that we have these three red blocks, three red specific promoter elements responsive to dehydration, and a yellow one, which is an additional specific one. And this yellow one is not present in the Lindernea brevidens, which is the middle one. And I told you it has a sort of intermediate phenotype, and the yellow stands for dehydration responsive element in the promoter. So that is missing here, and that explains why this gene is slightly less expressed than in Lindernea brevidens than in Craterostigma plantageneum. And if you go now to the desiccation tolerant plant, the Lindernea supracemosa, you see that only two of these promoter elements could be identified, and that clearly is not sufficient to get a high level of expression. So the conclusions from these comparative promoter studies are that conserved promoter elements, which are responsive to dehydration and to the phytohormone abscisic acid, determine the promoter activity. And we found most of these elements in the Craterostigma. And Craterostigma is desiccation tolerant in its native habitat. So I think that could be important. So it experiences water deficit throughout its life cycle several times. And Lindernea brevidens, I told you, is the plant which comes from the rainforest. And one possible explanation, but we cannot prove it, is that therefore it has perhaps even lost some of these essential promoter elements. So it has fewer elements, but it still can be upregulated. However, Lindernea supracemosa is only very slightly expressed and has more or less lost this desiccation tolerance and lost the ability for a high induction of the promoter. So my hypothesis to this is that mutations also in the promoter region, in the regulatory region, are not random, but they are driven by the environment. And this has been supported by two different recent analyses in different organisms, and the citations are written here on the slide. So now I would like to turn to another group of proteins. So I told you that the resurrection plants have the problem that, I mean, not all of them, there are two different groups, but the groups of resurrection plants with which we work, they retain part of their chlorophyll. So they still absorb light while they're sitting there in the desiccated state. And we heard that already yesterday from Professor Hense, that when these plants are sitting there, they can be attacked by pathogens, but they're also attacked by light, if I may say so. So what do these plants do? They have a high expression of photoprotective proteins. I mean, again, this is a bit of a hypothesis that these proteins are really photoprotective. The true biochemical proof has not exactly been given yet. And this group of proteins are the so-called ELIP proteins. ELIP were actually discovered when somebody studied the germination of barley. And ELIP stands for early light-inducible proteins. So as soon as there shines some light, these proteins appear. And again, that is also a process which needs some protection from the full sunlight when you think of the germinating seedling. And already when we studied this grass, the Oropetium, we found something quite remarkable, which I will show you. Namely, that this ELIP gene in all desiccation-tolerant plants occurs in very high copy numbers in the genome, and only in a few copy numbers in non-tolerant plants, for instance, Arabidopsis or also wheat or barley, only 2 or 3 copies. And what we further observed, already studying studies on the Oropetium, were the overrepresentation of these ELIP transcripts in leaf tissues, similar to the Lea proteins in the desiccation-tolerant plant. So only one short example here without going into detail. So just to show you that we really studied it. First of all, take a look at the bottom of this slide. And I've written some numbers there. In the Cratostigma plantageneum genome, we find more or less nearly 200 copies of this gene. In the Lindernia brevidens, we find something like 180 copies. However, only 4 copies in the Lindernia supracemosa. And how can we explain, and that is sort of tried to depict on the left-hand side, these different copy numbers in the genome. So it seems that during the evolution of desiccation tolerance, there have been several independent events of tandem duplications of these ELIP genes. I mean, we monitored more or less all the sequences of the desiccation tolerant plants which are known, and we found that they're overexpressed in all of them. And here on the right-hand side, you see an expression analysis during dehydration, comparing the two Lindernia species. Again, you see, if you go to the left-hand side, which is Lindernia brevidens, you see that it's more brighter spots, and that means higher expression. Just to show you that we have evidence for an overexpression or high expression of this gene under dehydration, very finely tuned in these different stages during the dehydration, and then a certain appearance in the rehydration. So overrepresentation of these ELIP transcripts in leaf tissues was reported in Cratostigma plantageneum and in Lindernia brevidens, and also, as I just mentioned, in all the other desiccation tolerant species for which genomic sequence is available. And this can be explained by gene duplications, which certainly contribute to the higher abundances in these different species. And with the numbers, I think they are very impressive, going from 200 to 4. So coming to the end of my talk, more or less. So many data are available which describe the changes which take place during dehydration, rehydration. I have given you some examples. I have pointed out that there are some protective proteins which are expressed during the dehydration process. I have not shown you that they also disappear during the rehydration. So there are 2 mechanisms which lead to this high expression. I have given you evidence for selective promoter elements which regulate a higher expression, but that is not only sufficient, it also requires, obviously, duplication of genes during evolution. And the big question is, which are the signals which trigger these processes? So we have done, and I have no time to show you this, some experiments looking at the cell wall, and I told you that there is a shrinkage of the cell which leads to mechanical stress, and we see changes in the cell wall composition, and we also see changes of potential signals, which includes a kinase, which we have studied in more detail. However, I also would like to point out here, and maybe this is still interesting, although many people try to do something, that the true biochemical functions of, for instance, these layer proteins, and to some extent also true to the ELA proteins, is not really fully understood. So there's still quite a bit of work to do. What could be the benefits? Coming back to the question which I asked in the beginning, from studying these desiccation tolerant plants, why is it not only a luxury to do this work? It is possible, and if I have time, I will show you a very short slide. Identification of mechanisms to withstand water deficit in agricultural important plants. I mean, we don't have the illusion that we want to make a wheat plant and a resurrection plant. That will not be possible because we don't have, for instance, the evolutionary processes which are necessary. We heard yesterday, a benefit of these studies are the discovery of interesting metabolites. I mean, I just listed 3 here, which we have identified and studied a little bit. This is a compatible solute, a glycerol derivative. It's antioxidant verbascoside, which is a phenolic compound. It's also a family. And I have mentioned in the introduction also this unique occurrence of the CH sugar, the octalase, which also may have some antioxidant function, but that is probably not the only one. And then I think it could be a very attractive to try to understand what happens really under the desiccated state in these cells, what reactions are possible or what are not possible, triggered by the changes in the protein conformations. But that is something for the future. I don't know, do I still have time, a couple of minutes, or yes, no? Okay, very quick. I just want to give you one example because I've been talking about are there some immediate benefits? Can we isolate some genes which are useful for improving stress tolerance? And I give you one example. It's an enzyme, an aldehyde dehydrogenase, which contributes to the abundance of redox equivalents. And we overexpressed this gene in Arabidopsis, and we could show that these transgenic Arabidopsis plants make this protein. It's an enzyme, and here you see these resulting plants. On the top row, you see the transgenic plants. From left to right, it's the control, then a dehydration experiment. And if you go to the bottom, these are the non-transgenic plants which don't contain an additional copy of this gene. And you can see that apparently the addition of one more gene of a family seems to provide a certain level of tolerance. And just to summarize some of the highlights which I wanted to relate to you, namely that there is a fine-tuned genetic network which enables desiccation tolerance. It's not one gene, it's not a miracle strange gene, but it's a genetic network which is necessary. There is a core set of desiccation responsive genes. We also observe some differences if you look into the root, into the leaf tissue. So also that requires some more detailed studies because they have different functions, roots and leaves. And I think I emphasized a lot that gene duplications and selective promoter elements are really some essential evolutionary drivers towards establishing desiccation tolerance. And I've sort of indicated what could we learn from these studies, what are possible avenues to follow. And in the last slide, I would really like to acknowledge the collaboration with Rob van Buren and his team at the Michigan University, which was essential for all the genome analysis. Without him, we couldn't have done it. Eberhard Fischer from Koblenz collected some of these related plants and provided them to us. And we had various collaborations and, of course, many collaborators, some are even active in Bayer, have contributed towards the story. Thank you very much for your attention.
Thank you very much for this really fascinating talk. And what we learned is that if it's about coping with stress, it's not just about having exotic mechanism. There's also a simple sentence, more is more. And starting with this, it would be now great to have some interesting questions from the auditory. Yes, here you are.
Yeah, thank you very much. My name is Ralf Nau, and I'm insect toxicologist. I'm not a plant physiologist by training, but I very much enjoyed your presentation. And I noticed that the differences are not so extreme between plants and insects in terms of adaptation to environments.
True.
We also have gene duplications. We have observed a certain synteny for gene groups and also tandem repeats driving the overexpression of such genes. It's really interesting. I have one question regarding the LIA genes you have shown. And you mentioned that you also may have observed a kind of neo-functionalization of these genes by gene duplication. Have you been able to check what has been changed? Is it a structural change which make them more resistant to survive in a waterless environment?
Well, I cannot completely answer your question. So it may be an indication towards more hydrophilicity. But this needs to be studied in more detail. And we have got together now with Guido Grossmann from Düsseldorf University, and they're cell biologists, and they would like to look into the cell and really follow different layer proteins, what happens in the dehydrated state, and which motifs are essential. So maybe this doesn't really answer your question, but we don't know. But as I also mentioned, I mean, insects have something in common with plants. I told you there are also some desiccation tolerant nematodes, so they're not insects, but still they have to, yeah, they seem to have a similar mechanism. I mean, okay, they have also invented the layer proteins.
One further question? You had one?
Thanks a lot. You really woke me up after lunch. This is really helpful. Thank you. So I have maybe two special questions for the layer proteins again. So you showed this the study by Hunt et al. From 2011. Mm-hmm. I guess these were simulations just from the pictures, or were these experimental data you showed us for the different?
No, they're simulations. I think, but they also also did some experiments to try to determine the structure. I mean, I think the first slide was actually taken from a review, which was still much older, and they made structure predictions. But
yeah. And now my actual question is: have you tried out what these AI-based folding things, alpha, open, ESM, whatever fold, how they deal with these LIA sequences?
No, we have not done that.
You should.
But, okay. Well, there's only a limited number of experiments one could do, and one does these experiments, you know, which are the most easiest to do. For me, the molecular experiments are the most easiest to do. So, but this also shows you, and I think this has been one thing I learned during my whole career, is that if you collaborate with experts, you get more out of it. I mean, the collaboration with Rob van Buren was fantastic, is fantastic, and others, I mean, with structural biologists and so. So maybe that's a message also to the youngest.
And now it's time to say hello, St. Louis. Did you enjoy the speech as we did? Is there any question from St. Louis? Hello?
Hi, yes, hi. Sorry. This is Graziana. So I was wondering, thank you for the talk. I was wondering for your ELIP proteins, the one that are, okay. Hi. So for the ELIP protein, you mentioned that you have more in the resurrection plants. Are they duplicated? Is the duplication in tandem, and you think they are co-regulated, some sort of operon, or they are just you have 200 across the whole genome compared to only 4 in the non-desiccated?
Sort of clustered.
They're all
But
clustered. Okay.
But it's not all in tandem. I mean, as I told you, this cratostigma is octuploid. It has actually 56 chromosomes. So if you try to align them, you find these duplications in several of in most chromosomes.